Genetic patenting. In the United States, 220,000 women are diagnosed with breast cancer each year. Medicine's latest conundrum is cracking the code of what we've come to know today as cancer. As we all know, cancer is an umbrella term for what occurs when a cell is mutated and loses the ability to stop replicating, creating malignant tumors that have varying consequences on one's health. Researchers all over the world are tackling this issue head on. However, it wasn't until recently that a huge obstacle was overcome in the United States. That obstacle is gene patenting. In order to fully understand the field of gene patents today, we have to look back 15 years ago when the human genome was mapped. The mapping of the human genome brought closer the world of law and the world of science. As legal experts saw potential in bringing the concept of ownership to genetic code of the human genome, about 20% has already been patented today. But the rules have changed since the first gene patent in 1982. To patent something means to claim ownership over an idea, design, or concept. Typically, the purpose of patenting is to protect the original owner of the idea, design, or concept to ensure that they are paid in if a third party wants to use their idea. Gene patenting means to allow unique segments of DNA which perhaps code for a certain disease or a certain protein to be owned by an individual or corporation. Gene patenting has both positive and negatives. There is a lot of misunderstandings with patenting genes. A lot of people seem to think it is harmless on the surface, but really when coming to curing cancer and diseases, it is conflicting with the medical community. Back to breast cancer. In the 1990s, Myriad Genetics, an American molecular diagnostic company, found that BRCA1 and BRCA2, a human gene that produce tumor suppressor proteins, with either of these two genes are mutated, then the female with this mutation will have a 20 to 25% chance of having hereditary breast cancer. Myriad wanted to then patent these genes that they so-called found, which would make sense, but in reality, that would mean no other company would be able to do research for these exact genes. And that exactly happened in 2000. So they pretty much started a monopoly on these genes that testing for these genes. So if your mom had breast cancer and you wanted to find out if it was hereditary, you would get the BRCA1, BRCA2 test. Yet the only company offering it, because they own the patent for it, is Myriad, and they are offering the test for $5,000. Another downside, besides the high cost, is there's no test to compare the results to. So there's a high possibility for false positives or false negatives because there's no way to double check. The positives to gene patenting is that smaller molecular biology companies, if found, say, the gene for Alzheimer's, would get the chance to make money before big companies would override them. In the 1980s, Congress passed a law, the Bayh-Dole Act, which encouraged universities to patent, which pushed federal finances to go more toward biomedical research. The National Institute of Health, one of the world's foremost medical research centers, funding went from $5 billion to $26 billion from the 70s to 2003. Another positive to gene patenting is that all the jobs it has created over the years. Also in 1980, a Supreme Court case called Diamond v. Chukabarka ruled that man-made living organisms could be patented. This explains why Myriad was able to patent the BRCA1, BRCA2 gene in the 90s. Fast forward 30 years later, another court case came up, the Association of Molecular Pathology versus Myriad Genetics. The Supreme Court ruled in 2013 that a naturally occurring DNA segment is a product of nature and not patent eligible merely because it has been isolated, but determined that cDNA is patent eligible because it is not naturally occurring. So pretty much just separating the gene from its surrounding genetic material is not an act of innovation, just a finding. But with cDNA, which is DNA copy synthesized from mRNA, which is all done in the lab, so even though the cDNA is a piece of a patient's real DNA, because it is synthesized and all done in the lab, it is considered unnatural. So patentable. Enter 2015. Gene patents and ethics still come in contact with each other often. There are three main ethical concerns that surround gene patenting. The first argument views the patenting of genetic material as unethical on grounds that it treats life as a commodity. The second views living material as natural and therefore cannot be patented. And lastly, a segment of the population fears that allowing patents on genetic material will undermine the dignity of people and other animals through subjecting their genes to ownership of other people. Next to ethical concerns are concerns regarding consent. When it comes to gene patents, consent is required in three forms to be considered valid. 1, from the individual owner, 2, on behalf of the national level, and 3, consent on behalf of the community's level. In other places around the world, the European Patent Office ruled that patents cannot be filled if they revolve around the destruction of human embryos or human embryonic stem cells. In 2014, Australia rules that isolated genetic material is in fact valid subjects for patents. Japan, meanwhile, requires that patents innovations have to be marketable or have commercial potential, not including methods of surgery, therapy, or diagnosis of human diseases. Gene patenting is a complicated issue in the world today. The different favors regarding on the rules and regulations vary depending on your longitude and latitude coordinates, and the ethics are often viewed in shades of gray. One thing is for certain, it has come a long way since the 1980s.